histology of the cerebral cortex. In this section, you are seeing a coronal section of the brain in which you can see the outside is the cerebral cortex, which is arranged almost like a ribbon that is investing the cerebral surface, the surface of the cerebrum, right from above the corpus callosum to this point. So inside that you have the white matter. This is gray matter. Gray matter on the outside is called cerebral cortex, as I mentioned earlier in a video. And you have also masses of gray matter within the core of the brain, which is not called cortex. They are called nuclei. For example, this is the largest nuclear mass, which is the thalamus. So this is the cerebral cortex. And if you take the cerebral cortex in a histological perspective, the cerebral cortex is basically gray matter, and that means that contains cell bodies, that contains a high proportion of cell bodies. So these are the cell bodies that are stained here with a special stain. This is called Nissl staining method, where cressyl violet is used. And you can see the cell bodies are shown here as closely opposed dots. So all these dots are cell bodies of individual neurons. So that will create this dark color on the outside. That is, this gray matter is seen in a histological perspective. It is seen like this. On a careful examination, you can see that this dark layer is actually containing multiple layers. It has a laminar architecture. That is the important word that you need to remember. Cerebral cortex is arranged in a laminar architecture, and you need to know what this laminar architecture is. This is the coronal section. So what you are seeing here are all sulci, and these are gyri. You can see that this cortex is basically seen on the outside of the gyrus, but also on the walls of the sulcus. That is also important. Here you can see the wall. This is a large fissure, but here also you can see walls of the sulcus. Actually, the cortex is dispersed. It is arranged 1/3 on the outer surface and 2/3 is arranged on the walls of the sulcus. So when you look at the outside of the cerebrum, you are actually seeing only 1/3 of the cortex. But if you sort of strip or you separate all the cortical ribbon, if all the sulci are flattened, then you will get the entire picture of the cortex, and it is much more. The 2/3 is hidden within the sulcal walls. So you can see that clearly in this section images. 95% of cerebral cortex is 6-layered, and that is called the neocortex, the evolved cortex. But there are some regions of the brain that are also important, that are the archicortex and the mesocortex. That I'll consider in the end of this discussion. So we'll jump directly into the neocortical architecture. So this is the neocortical 6-layered arrangement. As I mentioned before, this is a Nissl staining method. This is actually not looking colorful because this is an excerpt from a paper that was in the early 1900s. What is Nissl staining? Nissl staining is a special staining method where the cell bodies of neurons are stained. The dye that is used is a basic dye, basically cressyl violet or toluidine blue can also be used, but it is basic. The dye is basic. So that dye will come and adhere or absorb towards acidic parts within the cell body. And the acidic compounds in the cell bodies are the DNA within the nuclei and the RNA within the cell body cytoplasm. You have the rough endoplasmic reticulum, which is called the Nissl substance, if you remember. That these are the substances that will be adhered by a basic dye. So basically the Nissl staining is used to visualize the cell bodies. That's the point that I want you to remember. So when Nissl staining is used to visualize the cell bodies of the cerebral cortex, you can see 6-layered laminar architecture because of different types of cells in each layer. These 6 layers are named using Roman numerals, and we'll come to the names of each of these. The outer one is called molecular layer. Why is called molecular layer? You can see that the outer layer is cell spares. The cell bodies are very, very less. You have a similar molecular layer in the cerebellum also, where also that layer is cell spares. Just deep to that, you can see the second layer, which contains a lot of dots. These dots are cell bodies again, neuronal cell bodies, and this is called the outer granular layer. So basically a cell body, it has a few dendrites in the surrounding, branching towards the surrounding region, and a few axons going close by. So that they are called granule cells. Deep to that, you can see the third layer, where you can see, in spite of granule, you can see granule cells there also, but the more catchy point that you see here are isosceles triangles. They are the pyramidal cells. Why are they isosceles triangles? Basically, these cells in a 3-dimensional perspective, they are conical. Suppose this is the base and this is that cone. Okay, the apex of that cone will give off an apical dendrite. Okay, the apical dendrite will be going superficially towards the pile bar, towards the pile surface. So all these apical dendrites will be directed superficially. That is why all of them are looking characteristically like isosceles triangle. On the basal aspect, on the periphery of that cone, you will have basal dendrites also. So you have an apical dendrite and you have basal dendrites. Now from the basal surface of that cone, you will have an axon that is descending downwards. So unlike the granule cells which had dendrites and axons in its vicinity, here the apical dendrite will go in the superficially, the basal dendrites will be in the vicinity, but the axon that arises from here will penetrate, which will classically penetrate the deeper layers. So these two cells are the most important cells that you need to learn in the cerebral cortex. The other types of cells are horizontal cells of Cajal, the cells of Martinotti, fusiform cells, but these two are the most important. So deep to the third layer, you have layer 4, and layer 4 contains granule cells, again similar to the layer 2. So this is called the inner granular layer. And deep to that you have again pyramidal cells, but you can see these pyramidal cells are in stark contrast to this. These are much more larger. Okay, in specific areas of the brain these are very large. Example, the precentral gyrus, these cells are huge. If you take a comparison of size, these are granule cells are approximately 5 to 10 microns. Okay, this will be 10 to 40 or 50 microns. But these cells, the large pyramidal cells, especially in the precentral gyrus, they are humongous. They are 100 microns in size, and this will form one of the largest cells in the entire nervous system. These are called the giant pyramidal cells or Betz cells, B-E-T-Z, Betz cells. Now deep to the inner pyramidal layer, you have a polymorphous layer. If you look carefully into this layer, you can see different sizes and shapes for this cell bodies. So that is why it's called polymorphous. Poly means many and morphous means shapes. So these are the 6-layered classical arrangement of the cerebral cortical layers. Now this picture is showing a few scientists who researched on this layered architecture of the cerebral cortex. This is the head of that scientist group, and this person's name is Vogt. This is his wife. This is Oscar Vogt, and this is Cecily Vogt. This, what you have learned now, is the cytoarchitecture based layer arrangement. But these two persons, the couple Vogts, they actually researched on another aspect called myeloarchitecture, where you are not learning about the cell bodies, but you are learning about the fibers. So that is this map. This is based on another staining method called Weigert stain, where myelin is stained. So myelinated fibers will be stained in Weigert staining. In that you can see two sorts of arrangement. One arrangement is like this, tangential to the pia mater. Okay, so that sort of arrangement is seen, as well as radial, that will be penetrating through the different layers. And here you can see again 6-layered pattern, which is almost corresponding to the cytoarchitectonic map. The cytoarchitectonic map is classically described in Roman numerals. The myeloarchitectonic map, to avoid confusion, that is written in Arabic numerals. But both of these are almost similar. So in the myeloarchitectonic map, I want you to know two important tangential fibers. They are fibers that are present in the layer number 4 and the fibers that are present in the layer number 5. So these two layers are the external band of Baillarger, and this is called the internal band of Baillarger. The external band of Baillarger will be in the layer number 4, and the internal band of Baillarger will be in layer number 5. Okay. The cytoarchitectonic map was extensively researched by this scientist, and this scientist is Corbinian Brotman, the German scientist who parcellated the entire brain into 52 cytoarchitectonic areas, and that is called the famous Brodmann's area, and he rose into enormous fame unlike the Vogt couple. So I want to mention one more point. Where are these cells going and connecting into? So from the layer number 6, you will have fibers that are projecting to the thalamus. We call these fibers corticothalamic. The layer number 5 will have fibers that are projecting to subcortical regions other than the thalamus. So 5 and 6 are projecting to subcortical regions, 6 is to thalamus, and 5 is to regions other than the thalamus. Example, the corticospinal fibers to the spinal cord, the corticobulbar fibers, the corticostriatal, all this are coming from layer 5. What is the importance of layer 4? Layer 4 receives inputs. Most of the afferents to the cerebral cortex will be received by the layer 4, especially the sensory inputs coming from the thalamus, the visual input, the auditory, the somatosensory, vestibular, all of them will be projecting to the layer 4. So if you are taking a cortex that has a particularly increased number of sensory inputs, layer 4 will be much more thick. Layer 2 and 3 will be containing fibers that are connecting to other cortical structures, other cortices. 2 will be containing short association fibers, the 3 will be containing long association fibers, the 3 will also be containing commissural fibers. So 2 and 3 are basically interconnecting to other cortical areas. So all these fibers will come and branch in layer number 1. So this is the basic laminar architecture. With this idea, I think we can have a look at one of the virtual slides. This virtual slide, I have to thank Dr. Michael Horsch from the University of Michigan, who gave this set of virtual slides which have been used for us to teach histology in a much better way. So this is a gyrus. You can see a gyrus and sulcus. Now here you can see in the periphery, you can see the vascular pia arachnoid. This is the pia mater and this is the arachnoid matter. And within the arachnoid, you can see very beautiful blood vessels. Okay, these are all blood. You can see the RBCs over here and the vessel wall. So that is the pia arachnoid, the vascular pia arachnoid, and you can see the pia mater over here, and you can also see blood vessels penetrating through the pia mater. Okay, deep to that you can see the layers which I have marked over here for clarity. So you can see the outer layer here, that is cell spared, that is a molecular layer. Deep to that you can see a region where you have more granule cells. Okay, this is a granule cell, this is a granule neuron, and between that you have very small dot looking nuclei, naked nuclei. They should be oligodendrocytes or glial cells, mostly oligodendrocytes over here, which will be helping in myelination. Now deep to that you have the outer pyramidal layer. You can see a pyramidal cell over here, you can see the classic isosceles triangle look with the look over here. Okay, you have basal dendrites here. This is the apical dendrite. The axons, the distal axons will be taking the myelin stain. I forgot to mention the stain here. This should be a Nissl as well as a Luxol fast blue. Luxol fast blue will be staining all the myelinated fibers, and you can see the thick blue within the core of that gyrus, because that will all be containing white matter, but this will be cortex. And you can understand in this picture that cortex not only contains cell bodies, it also contains fibers arranged radially as well as tangentially. You can see the tangential arrangement of blue lines. Okay, that's showing the myelo architecture. This was a second layer, the outer granular. Deep to that you have the outer pyramidal. Deep to that you again have inner granular, and deep to that you have the inner pyramidal. You can see the inner pyramidal has larger sized pyramidal cells. Okay, when I say large and small, you can compare. You can see this pyramidal cell and compare that with this pyramidal cell. You can see how larger this pyramidal cell is. Okay, you can again see the isosceles triangle like look, the cell body, the nucleus with the nucleolus inside. This is the nucleus and this is the nucleolus. Okay. And deep to that you can see the polymorphous layer, which contains different shaped cells. Okay, deep to that you can see the white matter. And if I zoom into the white matter, you can see the axons as well as nuclei that are present. These nuclei should be the glial cells that is going to myelinate these axons. So that is the arrangement of the cerebral cortex. In a histology spotter exam, you may see a field like this, and you can identify it as cerebrum by identifying this classical isosceles triangle looking pyramidal cells that will be directing towards the pia mater. But you will see pia mater only if you have an entire section of the cerebral cortex, which may not be present in some of the exams. You will have only a bit of the cerebral cortex like this. Now, if you look into another slide, this is again a slide of a particular region of the cerebrum, and if you look at that, you can see a blue line that is looking horizontal. So this is that external band of Baillarger, and this external band of Baillarger in a particular region of the cerebral cortex is extensive, and it is so thick, and that region is in the visual cortex, and that was described by a person called Francisco Gennari, who was a medical student in 1776, and he described it in a gross anatomy specimen, and he showed that that cerebral cortex in that particular region has a white stripe, and that is called the stria or the stripe of Gennari. And this is the reason why this is called striate cortex. Why the stripe of Gennari is only there in here? Because that is an extensive, dense projection of optic radiation from the lateral geniculate body of the thalamus. The lateral geniculate body projects onto the striate cortex. So that is an extensive, a dense projection into the striate cortex, which will create an extensive band of Baillarger, external band of Baillarger, and that is called the stria of Gennari. So this is macroscopic. I'll show that in a specimen in the brain and the occipital cortex section in further videos. So that was in 1776, but now after 250 years, in 2016, Neuroimage published a paper by Fracasso in which the lines of Baillarger and the stria of Gennari was displayed in a 7 Tesla MRI, and which was correlated histologically. You can see band of Baillarger is shown as a blue line. Again, with Luxol fast blue, you can see it as a blue line, and that has been correlated with 7 Tesla ex vivo and in vivo imaging, and in which he has shown within that sulcal, within that calcarine sulcus, he has shown that stria of Gennari and as well as the bands of Baillarger in the extrastriate regions. So this shows the importance of knowing this histology in wide regions, including neuroimaging research. In anatomy exams, usually there is a question, what are the other types of cerebral cortex you know of? I told you the 6-layered cortex is called neocortex. Neocortex is further classified into granular in sensory cortices. I mentioned the layer number 4 and 2 will be much more elaborate in sensory cortex. Example, visual cortex 17, 18, 19. Also auditory cortex area number 41, 42 and 22. The sensory cortex area number 3, 1, 2. All these are sensory cortices on a broad head, and they will all contain granular regions much more developed than these pyramidal regions. So they are called granular cortices. But if you take motor cortex, example the precentral gyrus or the area number 6 or 8 or limbic cortices, those regions we will have granular cortices where these layers, the layer number 5 and the layer number 3 will be elaborate. The other regions will be dwindled. You also have a cortex called polar cortex where all the 6 layers will be diminished. The other regions of the frontal and parietal lobes, you are called as frontal and parietal cortices. This sort of classification may be asked in exams. So these are the types of neocortices. If you are asked the types of cerebral cortices, then the answer is different. They are the neocortex. Example, if you take temporal cortex over here, that is a neocortex. But if you take a cortex over here, okay, the temporal cortex, if I trace the temporal cortex from here, temporal cortex can be traced onto the inferior temporal lobe, can be traced onto a region over here. This is the parahippocampal gyrus, and then I can trace that further into a peculiar formation that runs like this, that is folded like this. This is called the hippocampus. If you take the hippocampus, that is not 6-layered. That is unlike this 6-layered neocortex. That is not 6-layered. It is only 3-layered. And that 3-layered hippocampus forms the archicortex. Neocortex is a highly evolved, evolutionarily advanced cortex, but archicortex is an evolutionary primitive cortex. So hippocampus and this one is called dentate gyrus. They are all examples of primitive cortices or archicortex. So you have other cortices which are a transition between the archi and the neocortices. They come under limbic cortex. Example, the parahippocampal gyrus, the cingulate gyrus. There you can see it's a trade-off between 3-layered and 6-layered cortices. So they are called mesocortex. So these are the other types of cortices that are that can be mentioned when you are asked the types of cerebral cortex. So with that, I think we can wind up the discussion on cerebral cortex. Thank you.